Biology paper 6 alternative to practical May-June 2024. Paper 62 alternative to practical. Okay, let's start. A student investigated the effect of acid concentration on the rate of diffusion in agar gel. The student was given 3 test tubes containing bromethamol blue indicator in agar gel. Bromethamol blue indicator in agar gel. Bromethamol blue is a blue indicator that turns yellow in the presence of an acid. The student used this method. Step 1. Label 3 beakers A1, A2 and A3. Put 20 centimeter cube of 1 mole per dm cube hydrochloric acid solution into beaker A1. Okay, now name 3 beakers. A1, A2 and A3. So put 20 centimeter cube of 1 mole per dm cube hydrochloric acid solution into beaker A1. So we put 20 centimeter cube 20 cm cube HCl. Put 2 centimeter cube of 1 mole per dm cube hydrochloric acid solution into beaker A2. So in beaker A2 we put 2 cm cube HCl. Add 18 centimeter cube of distilled water to the hydrochloric acid solution to beaker A2. So we have to add about 18 centimeter cube water. Use a clean syringe to transfer 2 centimeter cube of the solution from beaker A2 to beaker A3. So we take 2 centimeter cube from this beaker and add to this beaker 2 centimeter cube. Add 18 centimeter cube of distilled water to beaker A3 and add 18 centimeter cube of distilled water in beaker A3. Table shows the final concentration of the hydrochloric acid solution in each beaker. So, final concentration of the hydrochloric acid solution mole per dm cube, final volume of the solution 20, A3 20 and A2 is also 20. Next. Label the 3 test tubes that contain the bromothymol blue indicator in agar jelly A1, A2 and A3. So we take 3 test tubes. And it have bromothymol blue indicator. Bromothymol blue indicator. Use a syringe to transfer 10 centimeter cube of hydrochloric acid solution from beaker A1 to test tube A1. So we add about 10 centimeter cube of hydrochloric acid solution from beaker A1 to test tube A1. 10 centimeter cube solution. Use a syringe to transfer 10 centimeter cube of hydrochloric acid solution from beaker A2 to test tube A2. From beaker A2 to test tube A2, 10 centimeter. Use a syringe to transfer 10 centimeter cube of hydrochloric acid solution from beaker A3 to test tube A3. So also same with the test tube A3, 10 centimeter cube. Place the 3 test tube in a water bath at 35 degrees centigrade. So you have to place all of them in a water bath. A1, A2, A3 with all the solution. Start a stop clock and leave the test tube in the water bath for 20 minutes. After 20 minutes, remove the test tube from the water bath. Measure the temperature of the water in the water bath. Figure 1.1 shows the 3 test tubes after they were removed from the water bath in step 13. Okay. So if we see, this is blue guar jelly, this is yellow guar jelly in test tube A1, and this is the hydrochloric acid. A guar jelly containing bromothymol blue indicator. This is the height of yellow guar jelly. This is blue guar jelly in A2 and A3. आप इनकी हाइट का डिफरेंस देख सकते हैं. Measure the height of the yellow guar jelly in each test tube shown. Prepare a table and record the result of the investigation included in your table. Your measurements from figure 1.1, the final concentration of hydrochloric acid. Okay. So draw a table. Concentration, mole per dm cube, and this is height in mm. So write down the concentration. So we take the same concentration. One point zero zero, 0.10, and then 0.01. Now measure the height. The final volume of the solution in table 1.1 is 20. क्या आप देख रहे हैं इसमें जो तीनों बीकर थे आपके पास दे हैव 20. ठीक. So and we add 10 centimeter cube from each beaker to test tube. Now measure the height of yellow agar gel. So here we take measurements in mm, okay, millimeter में हम इसकी measurement ले रहे हैं and we put 10 centimeter cube of hydrochloric acid in each. So now measure the yellow agar jelly. So if you measure The height of yellow agar jelly, it is about 25 millimeter here, 14 millimeter here, and 5 millimeter here. So write it on table. Concentration in mole over dm cube. At concentration 1, height is 25 mm, plus minus, you can add plus minus. Here is 14 plus minus 1 mm. There is 5 plus minus. You can measure that height with your scale also. Centimeter को then convert to the millimeter. So 1 centimeter is about 10 mm. 1 centimeter is about 10 mm. Okay. And then if there is 10 centimeter, it is about 100 mm solution. State a conclusion of the investigation. Increase in the concentration of hydrochloric acid increases the rate of diffusion. Identify the independent variable and the dependent variable in the investigation. So the independent वेरिएबल में आपके पास आ जाती है concentration of hydrochloric acid, concentration of hydrochloric acid, and dependent variable में आपके पास आ जाती है height of yellow agar. Show the reading on the thermometer in step 14. So it is 20, 21, 22, 23. 23 degrees centigrade. Record the temperature of the water in the water bath at the end of the investigation shown in the figure. So temperature is 23 degrees centigrade. The student stated that the change in the temperature of the water bath was not a significant source of error in the investigation. Explain why the student was correct to this state. So the first temperature which was recorded in experiment was 35 degrees centigrade. And we place test tube in water bath for 20 minutes. So 20 minutes में उसका जो temperature है वो down किया, ठीक है? It is 23 degrees centigrade. So explain why the student were correct to this state. Because the change in temperature will affect all the test tubes. Change will affect on test tubes in water bath. So there is no error in investigation. Suggest a control experiment for this investigation. So for control experiment, we can use water instead of HCl. We can add water in agar jelly instead of HCl. The student repeated his investigation but left the test tube in the water bath for 30 minutes instead of 20 minutes. Suggest how the result for the second investigation would differ from the result for the first investigation. So we increase the time from 20 minutes to 30 minutes. So what is the effect? Height of agar jelly increases in all test tubes. Wood increases in all three test tubes. Okay, next. In an experiment, a student cut different size cubes of agar containing bromothymol blue indicator. The student placed the cubes in hydrochloric acid and measured the time taken for each cube to become yellow. The results are shown. Length of cube, volume of the cube, and the time taken for the cubes to become yellow. Complete table 1.2 by calculating the volume of 4 mm cube. So you have to calculate the volume of 4 mm cube. So it is cube. So cube जो all sides होती हैं these are so 4 So this is 64. Plot a line graph on the grid of the length of the cube side against the time taken for the cube to become yellow. We have to plot a graph. So we take time at y-axis, x-axis. So y-axis we take time taken. Time taken for cubes to become yellow. Okay. And y-axis we have length of cubes. Okay. So we take here time taken. We take the reading about 500, and from length we take it to the 10. So it is time in seconds. Time जो हम लेंगे वो seconds में लेंगे. So it is 0. Ten grids are equal to 2. 2, 6, 8, 2, 4, 6, here is 6, 6, 8, 10. 10. 1, 2, 3, 4, 5, 6, 7, 8, 9. Just 100. 200. 300. 400, and this is 500. Let's plot the graph. So what's our reading? These are 2, 4, 6, 8, 10, and 35, 60, 170, 275, and 445. 35. Just 10, 20, 30, and here is 35. This one. And 4 it is 60. 6. It is 60. And here. And then it is 170, 275, and 445. 170. Here is 170. 275. Not somewhat here. It is 275, and then 445. It is 400 and somewhat here. So we have to join that. Okay. Show on your graph how you obtain your answer. Use your graph to estimate the time taken for a cube with a side length of 3 mm to become yellow. So 3 mm is somewhat here. 1, 2, 3. 1, 2, 3, 4, 5. It is 3 mm here, and we take its length here. This is 3 mm. So its length is about 50. Then time taken. It is 50 seconds. 50. State two variables that should have been kept constant in this investigation. So one is temperature and another is concentration of acid. Temperature and other is concentration of acid. Question number 2. When a seed germinates, it grows into a seedling. Is a photograph of a seedling. Make a large drawing of the seedling shown in the picture. So you have to make a large drawing. जो आपने ड्राइंग बनानी है उसको आपने शेडिंग नहीं करना है, ठीक है। आपने उसको सिंपल रखना, बट आपने उसको लार्ज ड्राइंग बनानी है, ठीक है। वो आपको तकरीबन हाफ ऑफ द पेज को कवर करना है। So this is your drawing। थोड़ा सा स्मूथली लेकर जाते हैं। Now this is done। Line AB represents the width of the seedling. Measure the length of the line AB on the figure 2.1। It is about 31 plus -1 mm. The actual width of the seedling is 9 mm. Calculate the magnification of the seedling in figure 2.1 using the formula, and your measurement magnification is, so it is 31 divided by 9, which is 3.4. So it is 3.4. Figure 2.2 shows photographs of two different seedlings P and Q. Describe one visible similarity and two visible differences between seedling P and seedling Q. So, visible डिफरेंस जो है वो आपको सिमिलरिटी से वो आपको नजर आ रहा है इसमें. Root and shoot. They have both root and shoot. Seed coat, ठीक है, तो वो visible है. So we have to write root, radical, seed coat, seed covering present in both. Differences पे आ जाएं. We have to describe two differences. Difference number 1, P. P makes darker seed coat, while Q has lighter one. Difference number 2, shoot and plumule visible. Q has no shoot plumule. You can also describe about leaves. P has leaves, Q has no leaves. Then P has oval shaped seeds, Q has round seeds. So there are many differences. Which one you want to write, you can. As seedling grows, they change starch into reducing sugar. Describe the method you would use to test a sample of seedling for starch and reducing sugar. So you have to write down starch test with iodine and Benedict solution for reducing sugar, which is starch test with iodine, and this is Benedict test. So I can write for you both of the tests. Test for starch. Boil seedling in water to remove chlorophyll. Remove and place in dry paper. Add iodine solution. If starch present, it gives blue-black color. Then reducing sugar test, Benedict test. Grind seedling, make solution, add Benedict reagent in about 10 ml of solution. If reducing sugars are present, it will give red to orange coloration.